Good morning, everyone. I'm Serene Markosian. I'm the lead of the Assay Guidance Manual program at the National Center of Advancing Translational Sciences. I would like to welcome you to the first and inaugural webinar of the new webinar series that we've launched this year on preclinical translational science. I speak on behalf of the AGM team and all my colleagues at NCATS to share our enthusiasm in hosting Dr. William Kaelin as our inaugural speaker for this webinar series. Dr. Kaelin is the Sidney-Farber Professor of Medicine at Harvard Medical School and Dana-Farber Cancer Institute. He's also a senior physician in medicine at Brigham and Women's Hospital, and also has been a Howard Hughes Medical Institute investigator for decades. He obtained his undergrad and MD degrees from Duke University and completed his training in internal medicine at the Johns Hopkins Hospital. Dr. Kaelin has served on multiple boards and advisory boards, including the National Cancer Institute Board of Scientific Advisors and the AACR Board of Trustees. He's also a member of the National Academy of Sciences. He's won multiple prestigious awards, including the Albert Lasker Basic Medical Research Prize, and most recently, the 2019 Nobel Prize in Physiology and Medicine. Dr. Kaelin's research seeks to understand how mechanistically mutations affecting tumor-suppressor genes cause cancer. And his long-term goal has always been to lay the foundation for new anti-cancer therapies based on the biochemical functions of such proteins. His most recent work has set the stage for the development and approval of mutant IDH inhibitors. His talk today emphasizes the importance of rigor, robustness, and reproducibility in research, particularly in preclinical cancer target validation. And this topic is actually very close to our hearts, and it perfectly aligns with the mission of our AGM programs. Hence, I'm extremely excited to have Dr. Kaelin as our inaugural speaker today. So before I give the stage to Dr. Kaelin, I'd like to let the participants know that they can ask their questions to the Q&A chat box, and we will get to the questions at the end of the talk. And then if you think any of the questions is of interest to you, you can feel free to vote those questions so we can get to them before others. So with this, thank you so much, Dr. Kaelin, for joining us today. The floor is yours.
Thank you very much for that very nice introduction. Okay, well, thank you very much. It's really a pleasure to be with you virtually. Here's my disclosure slide. The most relevant one for the talk today relates to a HIF-2 inhibitor called belzutifan being developed by Merck. The title for this talk, in part, is a ode to this book called How Not to Be Wrong, The Power of Mathematical Thinking, which I like to recommend to young people. Although it's called The Power of Mathematical Thinking, it's really the power of logical thinking and the importance of thinking logically. And he gives a number of wonderful examples from various walks of life, including science. So this talk grew out of a growing appreciation that many times, in fact, the majority of times, when we publish findings in the academic literature related to new cancer targets, our friends and colleagues in industry tell us that they have trouble reproducing the findings, or at least the findings are not very robust. And a number of pharmaceutical companies have published that over half the time, and sometimes it's as high as 80 to 90% of the time, they have trouble replicating findings from primarily academic labs related to new targets in cancer. And so this really highlights the problem of reproducibility and robustness. And this got so bad that it started to get picked up in the lay press. And of course, this led to questions about what are you scientists doing with these precious resources that we're giving you if there's really this crisis with respect to reproducibility and robustness. Now, the first time I gave this talk, which was a number of years ago back at Cold Spring Harbor, people immediately said, well, Kaelin, you're combining the reproducibility problem with the robustness problem, and that's true, and they're related, but I think we should at least agree on what robustness does or does not mean, because frequently people think robustness in the lab relates to, say, large differences or very big differences, and it's true that everything else being equal, large differences are probably more likely to be robust than small differences, but robustness really, in my mind, relates to the ability to withstand perturbations. So, for example, commercial airplanes and the internet are designed to be highly robust. Multiple things would have to go wrong before a commercial airplane would fail to function in the air, or the internet would fail to function. And in the laboratory, robustness really, again, means the ability to withstand perturbations. So, for example, if a result was true, but it was only true at pH 7.35, but it wasn't true at pH 7.4 or 7.3, that begins to suggest maybe it's not going to be very robust. Or if the result is true, but it's only true in a particular cell line, it may not be very robust. A result might be true, but if it's only true when the Red Sox have won the night before and you went out celebrating, that's unlikely to be very robust. So we really want to come up with findings that are both reproducible as well as robust enough to withstand real world perturbations, such as in a clinical trial. Now, how do we get here? And my conjecture is one of the things that fed into the reproducibility and robustness problem is, I think there's really been a sea change in the past 20 or 30 years where I think young scientists feel pressured to justify everything they're doing in terms of its potential clinical relevance. And so I think people in their papers often, no matter what they've discovered, feel like perhaps in figure 5 or 6, they should have a figure that looks something like this, that sort of smacks of clinical relevance. So here I have IFG stands for the investigator's favorite gene. And so maybe in figure 6, they have some Kaplan-Meier curve where, at least in some tumor type, the expression of their favorite gene is associated with poor outcomes. And then maybe they've married that to a mouse xenograft study where they've knocked down their favorite gene and shown a decrease in tumor formation. So before I talk about the potential problems and pitfalls of these two types of experiments, I think it's useful to go back and go to sort of philosophy 101 and think about the ability to distinguish between correlation and causation. So if two things, we'll call them A and B, frequently correlate with one another, it could be that that's because A causes B. It could be because B causes A. Of course, both of those statements might be true. But often in biology and medicine, there might be a hidden confounder that causes both A and B. So here, A doesn't cause B and B doesn't cause A, but they're both under the control of a hidden confounder. And then finally, in the era of big data and multiple hypothesis testing, sometimes things simply appear to correlate, but it's really due to chance. If you test enough possible correlations, you're going to find some things that correlate, and it's just very important to properly correct for multiple hypothesis testing. And in thinking about a correlation versus causation, I think it's also important to understand the very simple concepts of necessity and sufficiency. So, for example, A is necessary for B implies that if you don't have A, then you can't have B, whereas A is sufficient for B implies if you do have A, then you will have B as a consequence. Now, part of the reason I go through that very simple exercise is I'm constantly surprised how many people, even at the professorial level, don't necessarily think in terms of necessity and sufficiency. So, for example, years ago, when BRAF activating mutations were found in malignant melanoma, almost immediately naysayers started saying, well, BRAF is not a good target in melanoma because BRAF mutations are also found in benign nevi, which parenthetically is a true statement. But the fact that you can find activating mutations of BRAF and benign nevi simply tells you that BRAF mutations are not sufficient to cause melanoma. Presumably you need other cooperating genetic events. But it didn't address the question of whether ongoing BRAF activity was necessary in melanoma cells. And fortunately, the answer to that turned out to be true. And of course, we now have a number of approved BRAF inhibitors for the treatment of melanoma. Now, maybe a more subtle example of failing to think properly about necessity and sufficiency is what I call the multiple mutation problem. So quietly, a number of people will say, or at least think, that real cancer, that is the common epithelial cancers, are just hopelessly complicated, and the cancer cells, because they have too many mutations, really will never succumb to targeted therapy. The implication here being, I suppose, if you have multiple mutations, you'll need multiple drugs targeting those various mutations, and quickly that becomes an insurmountable problem. But again, I think these people are not thinking properly about necessity and sufficiency. So to use an analogy, here's a lock, and of course you're familiar with locks, so you would know that it is necessary for all 3 tumblers to be in the correct position for the lock to open. But if I then asked you, well, then how many tumblers have to be out of position to keep the lock locked? I hope you wouldn't say 3. You would know the answer is 1. So I actually think it's a good thing that most cancers have multiple driver mutations. That tells you that those driver mutations were all necessary. And in principle, many of those mutations, when targeted, even in isolation, should lead to at least some measure of a therapeutic effect. And in fact, at least in the laboratory, and admittedly, these are somewhat artificial circumstances, at least in the laboratory, correcting even a single genetic defect in an otherwise genetically complex cancer can lead to a decrease in tumor growth, and in some cases, even a decrease in viability on a plastic dish. So this is consistent with the idea that even though cancers might have multiple driver mutations, each of those driver mutations, because they were necessary, presents itself as a potential therapeutic target. So now let's go back to the Kaplan-Meier curve. So the seduction on the left in this Kaplan-Meier curve is that somehow the investigator's favorite gene is causing this aggressive behavior. That's sort of the seduction, right? That this is actually causal, that the investigator's favorite gene is actually causing the bad outcomes. But just to show you how this is potentially fallacious thinking, let's take these fictitious Kaplan-Meier curves for people with chronic lung disease, where I've drawn the survival curves of patients with chronic lung disease who've ever needed to be on a ventilator versus those who've never needed to be on a ventilator. And certainly these curves would separate the way I've drawn them here. Needing a ventilator is certainly a biomarker for more advanced lung disease. But I hope you wouldn't conclude from this that the ventilators were necessarily causing the bad outcomes, and that we should immediately remove all the ventilators from the ICUs across the country because they're somehow killing our patients with lung disease. Now, that's a laughable example, but here's a more subtle example. I can tell you there are hundreds of papers in the literature in various cancers where it's been shown that tumors that have hypoxia, that measurable hypoxia or upregulation of the transcription factor HIF, which stands for hypoxia-inducible factor, it is certainly true that tumors that have marked hypoxia or have upregulation of hypoxia-inducible factor have a bad outcome compared to tumors that are not hypoxic and where HIF levels are low. Now, one explanation for that would be that indeed hypoxia upregulates HIF, and HIF causes aggressive tumor growth. But an equally possible explanation is that aggressive tumors outgrow their blood supply, become hypoxic, and therefore upregulate HIF. And so you can see that if you're thinking about HIF as a therapeutic target in cancer, knowing which of these two models is correct becomes really important. And of course, that you could come up with other explanations as well. Maybe hypoxia really does cause aggressive tumor growth, and it also upregulates HIF, but it's true and unrelated, that hypoxia is simply the hidden confounder that's causing both aggressive tumor growth and upregulation of HIF. So the punchline here is that a lot of times we take a shortcut in the literature where two things correlate with one another, and it's plausible that they're causative. And so I could come up with scenarios where HIF really does cause aggressive tumor behavior, but correlation plus plausibility is not yet proof of causation. You have to roll up your sleeves and do the hard experiments to see whether two things really have a causal relationship rather than really just correlating with one another. So if being associated with a bad prognosis doesn't necessarily make you a good target in cancer. How about being associated with a good prognosis? Maybe is it possible that being associated with a good prognosis disqualifies you as a cancer target? And again, I can tell you way back when IDH mutations were first found in various cancers, including gliomas, and when it was discovered that IDH mutations actually are associated with a good prognosis in glioma, I again heard professorial people say, well, mutant IDH is not a good target because it is associated with a good prognosis. In fact, I heard one professor railing that it would be medical malpractice to treat mutant IDH gliomas with an inhibitor of mutant IDH. But of course, these people, again, are confusing correlations versus causation. And just to show you, again, that this sort of logic is fallacious, really one of the best validated targets in all of cancer is the estrogen receptor in breast cancer. And yet it is also true that ER-positive breast cancers have a better prognosis than ER-negative breast cancer. But it is still nonetheless true that in ER-positive breast cancers, they depend on the estrogen receptor. So this is really an apples to oranges comparison when you really start to think about the different outcomes in ER-positive breast cancer versus ER-negative breast cancer. And so the conclusion here is that being associated with a poor prognosis is neither necessary nor sufficient for being a good cancer target. So I think there are lots of reasons to sometimes include Kaplan-Meier data in papers, but I think being associated with a poor prognosis is a fairly weak argument that something is a good target in cancer. So now let's move to the right and to the mouse xenograft assays that we frequently do. So these are problematic for several reasons. First of all, if you dive into the methods and really think about how these experiments are done, this difference that I'm showing you here on the right could be the difference between 10 cell doublings versus 9 cell doublings once the cells were injected into the immunocompromised mice and allowed to form tumors. So that in and of itself is already probably unlikely to translate into a very significant therapeutic outcome. But the problem I want to highlight today is that this is a so-called down assay as opposed to an up assay. So by down assay, I mean, here's some fairly complicated process. This happens to be a biological process, is now no longer happening quite as well as in contrast to, for example, an up assay where maybe some biological process is happening better, or where maybe some analyte is going up as opposed to going down. Now, people who do chemical and genetic screens, and I know we have a number of them on the call today, are familiar. In fact, it was them who really taught me over the years that people who do genetic screens and chemical screens prefer to do up assays when possible rather than down assays. And there are several reasons for that. So here I'm showing you a fictitious screen, and I'm showing you the signals on the y-axis and the samples on the x-axis. So first of all, up assays tend to have better signal to noise characteristics. It's easier to see a positive and a sea of negatives than the other way around. And of course, that's what makes astronomy work. But for our discussion today, the other reason up assays are preferable to down assays is you usually have fewer false positives or trivial positives because frankly, there are more uninteresting ways to make a complex system work worse than better. And so sort of the thought experiment, if you don't believe me, is to start removing parts from the internal combustion engine of your car and tell me how often the car performs better and how often the car performs worse. And this again gets back to necessity versus efficiency. The more things that are necessary for a biological or biochemical process, the more ways there are to interfere with it. And so whenever you do a down assay, there's this headwind of sort of uninteresting positives, technical positives. And in particular, it opens up the possibility that what you're really looking at is a so-called off-target effect or an off-target problem. And so just to formalize this, frequently in the laboratory, we add a perturbant to cells or mice. It might be a drug, it might be an siRNA, it might be a CRISPR guide. And maybe the drug or the siRNA has been designed against some target, and we measure a phenotype after we perturb the target. But especially when you're doing a down assay, you should worry that the effects you're measuring are really due to an unintended off-target effect. And so now I think I've introduced the ingredients that led to the reproducibility and robustness problem. I think in cancer biology and cancer pharmacology, we frequently add perturbants to cells. They're often loss of function perturbants, but the considerations would still apply to gain of function perturbants. We then do a series of down assays on our cancer cells that can be anywhere from decreased proliferation, decreased viability, to, for example, decreased tumor growth. And then, especially with the advent of high throughput screening technology, sometimes we get into issues related to multiple hypothesis testing. And then the cherry on top of the cake is we have a publication and promotion system that rewards so-called positive results. So maybe 10 people interrogated the same target, but the only group that got rewarded in the terms of a paper was the group that identified it as potentially being a good target. So and just to show you some real world examples of this now, our labs had a longstanding interest in the VHL tumor suppressor protein that forms a ubiquitin ligase complex that targets the HIF alpha subunit of the HIF transcription factor for proteasomal degradation when oxygen is present, whereas when oxygen levels are low or the VHL protein has been mutated, such as frequently happens in kidney cancer, now HIF alpha can accumulate, dimerize with its partner protein ARNT, and activate various and sundry HIF target genes such as VEGF. So a number of years ago, we determined that the likely problem in these VHL defective cancers, such as kidney cancers, was not only HIF alpha, but specifically HIF2 alpha, the less studied cousin of HIF1 alpha. So here's an early experiment. It's a tumor xenograft experiment, as you probably guessed. On the left, the cells were infected with an empty virus. And on the right, they were infected with a virus that included a HIF2 alpha hairpin. And in the interest of time, I won't show you the Western plots validating that this hairpin really does knock down HIF2 alpha very nicely. And lo and behold, you see that these cells lost the ability to form tumors, which was very exciting. Parenthetically, if you do this with a HIF1 alpha hairpin, you see the opposite result. So this was at least specific to HIF2 alpha. But again, this is a down assay. So lots of things have to happen correctly for these cells to form tumors. And now these cells are not forming a tumor, but could this be due to an off-target effect of this particular hairpin? Well, one thing that made us feel better was we could see this with multiple hairpins against HIF2 alpha. But really, the Cadillac experiment is to do a so-called rescue experiment, and that is to engineer or discover a version of your target of interest that's resistant to your perturbant. In this case, it was a hairpin, and see if you can reverse the phenotype. And so Keiichi Kondo in the lab did that, taking advantage of the degeneracy of the genetic code. He was able to engineer a HIF2 alpha cDNA, which he called MT star, that now still encoded wild type HIF2 alpha, but where the hairpin recognition site had been eliminated by virtue of translationally silent mutations, and that allowed him to do a rescue experiment. And lo and behold, indeed, now in the cells that had the HIF2 alpha hairpin, if they had this HIF2 alpha variant that was resistant to the hairpin, now the cells once again formed very large tumors, and that told us that this effect was indeed on target. Now, parenthetically, what things still keep me up at night? Well, first of all, I'm sure on occasion we're looking at things in the laboratory where the phenotype is a combination of the on-target effects and the unintended off-target effects of the perturbants. And if this is a so-called Boolean and, if you will, where both of these things have to happen, then the rescue will work because you now no longer have both arms here. But the point is, you might still be misleading yourself because you're still not recognizing the contribution of the off-target effect. And so here it underscores the importance of testing multiple hairpins, and frankly, when possible, having other corroborating lines of evidence, whether it's with CRISPR, whether it's with dominant negatives, whether it's with drugs, so you're not putting all your eggs in one basket. Another thing that keeps me up at night is that, and I've seen this over and over the years, almost invariably, consciously or unconsciously, the control that's used in these experiments has been selected to have no off-target effects. That, in fact, in many cases, they have no effects whatsoever. Or put another way, you don't remain in negative control very long if you have any if you have any deleterious effects on cellular fitness. So that introduces a bias where the control has been pre-scrubbed of any biological effects, and now you come along with your hairpin or your siRNA or whatever to your favorite gene. In fact, some companies, when you buy their control siRNAs, will proudly tell you that they've been empirically determined to have no effects whatsoever, which then introduces a bias. So sticking with now the role of VHL in kidney cancer, we had shown many years ago that if you restore the function of the VHL protein in VHL null renal carcinoma cells, they lose the ability to form tumors. Keiichi Kondo took these cells and introduced a version of HIF2 alpha that can't be recognized by VHL because of a point mutation in the VHL binding site. And these cells regain the abilities to form tumors. So this was an up assay. And I already showed you a moment ago the results with the hairpin experiments. And so this was nice because now we had both an up assay and a down assay. And now we can say something pretty strong. We can say that downregulation of HIF2 alpha was both necessary and potentially sufficient for the tumor suppressor activity of the VHL protein. Now, the conventional wisdom for many years was that HIF2 alpha wouldn't be druggable, but thankfully Rick Bruick and Kevin Gardner, who were then at UT Southwestern, identified a potentially druggable pocket in the so-called PAS-B domain of HIF2 alpha, which is what I'm showing you here. They then did high throughput screens at UT Southwestern and identified chemicals that could bind to this pocket, and in so doing, induce an allosteric change in HIF2 alpha such that it would no longer bind to ARNT and hence no longer bind to DNA. And these were out licensed to a company called Peloton that was then acquired by Merck, and I've already declared my financial conflict of interest. So the chemists at Peloton improved the drug-like properties of these compounds, and they were kind enough to provide to us a tool compound called PT2399 that was 1 or 2 atoms removed from what was then the lead compound. And Chin Cho in the lab, although Jim Brugarolas, my former postdoc's lab, had similar findings, we could show that in preclinical models, this compound did everything you wanted in models of VHL mutant kidney cancer. It decreased HIF-dependent mRNAs, it decreased proliferation, and it decreased tumor growth. So again, you want to declare victory, do high five, start writing the paper, but the operative word here is decreased, decreased, and decreased. So these are all more down assays. And again, we worry about down assays in terms of potential off-target effects. And so here we were lucky because Bruick and Gardner had already identified a point mutation in the HIF2 alpha PAS-B domain that would leave HIF2 alpha largely intact, but which would prevent the drug from binding to HIF2 alpha. And so what Chin Cho in the lab did was she used CRISPR to knock out the endogenous HIF2 alpha, which in these cells you can do, at least in the short term. And then she replaced the endogenous HIF2 alpha with an exogenous HIF2 alpha that was either wild type or had this drug-resistant mutation, or as an additional control, she introduced the empty vector. So here on the right are the Western blots. You can see the exogenous HIF2 alpha here. It also parenthetically has a V5 tag. NDRG1 is a well-studied HIF2 target gene. And I should point out that if anything, this point mutant is slightly enfeebled as a transcriptional activator. But that's a good thing, because if you introduce a bias into your experiments, it should always work against you rather than for you. So here we're going to try to do a rescue with a drug-resistant version of HIF2 alpha, but if anything, it's slightly impaired as a transcriptional activator. So just as an example, let's do a soft agar assay now with the Peloton HIF2 alpha inhibitor, PT2399. Here are VHL mutant kidney cancer cells expressing wild-type HIF2 alpha, and you can see at 0.2 or 2 micromolar, you suppress soft agar growth. Parenthetically, at 10 to 20 micromolar, this compound will kill all cancer cells, whether they express HIF2 alpha or not. So obviously concentration here matters. But I've said this is the kind of experiment that gives cancer pharmacology a bad name, because I could do this with Clorox bleach or any of a number of poisons if I titrated them appropriately. So how would I know this is on target? Well, I just told you a moment ago that Chin made isogenic cells where in the bottom, these cells have the drug resistance mutation, and now you can see these cells are insensitive to the drug. So this gave us great confidence that these effects were on target. Now, you might be saying we were cheating here because we were handed on a silver platter a drug-resistant mutation, and that's true. But I'll just tell you 2 technologies that we think, again, are highly robust for generating drug-resistant mutations of your favorite target when you're not so lucky. So one technology we like was developed by George Daly and Charles Sawyers, who were independently studying BCR-ABL and resistance to Gleevec. And what they did was you have a vector for your target of interest. but you propagate the plasmid in error-prone E. coli, such as XL1 red. So now the cDNA gets peppered with point mutations, and you introduce the pool into a sensitive cell line, treat with drug, and then you sequence the plasmid DNA from the survivors. And so this has worked very well for us. Frankly, though, the technology we're moving to currently to do this is to use current versions of CRISPR base editor technology. And so here we were studying an inhibitor of a glycosyltransferase called STT3A, called NGI1, developed by Joe Contessa's group. And so we designed a custom CRISPR library that would mutagenize the endogenous STT3A locus. And then we did an up assay for cells where STT3A was not inhibited. And we found a number of resistance mutations that were selected for in the screen that validated in secondary assays. So the nice thing here is you're dealing with the endogenous gene. And again, you can basically do saturating mutagenesis very quickly to generate your drug-resistant mutation. Now, as you should do, getting back to robustness, we did our experiments with the HIF2 inhibitor in multiple renal carcinoma cell lines that were VHL defective. And to our surprise, some were sensitive, as I told you, but some, frankly, were not. And you can actually see that if you go to the Broad Institute DepMap database and look at sensitivity to having HIF2 alpha eliminated using CRISPR guides. And so here, I'm showing you the kidney cancer cell lines in the DepMap collection. And all the cell lines in the bottom here were cell lines that depleted the HIF2 alpha guides over time in these pooled whole genome CRISPR screens, presumably because they're HIF2 dependent. But you can see, and we confirm that other renal carcinoma cell lines, despite being VHL defective, are nonetheless HIF2 independent. Now, and unfortunately, the editor initially said, well, you don't understand this heterogeneity, and so maybe your paper shouldn't be published yet until you understand this heterogeneity. But I'm old enough that sometimes editors answer my phone calls. And I said, well, don't you think that's part of the problem in publishing today, that a lot of groups might have been then tempted to write a paper focusing solely on the sensitive cell lines and left for another day the insensitive cell lines? It wouldn't be better to just honestly depict what we found and the fact that there's likely to be heterogeneity. And thankfully, I won that argument. But part of the reason I'm thankful I won that argument is, of course, this is exactly what was seen in the clinic. So here, what I'm showing you are swimmer's plots with a better version or more advanced version of the HIF2 alpha that's now called belzutifan. So each one of these horizontal bars is an individual patient with kidney cancer on the trial. For orientation, this is 1 year on therapy, so the x-axis is time. And these patients had been heavily pretreated and had failed at least one VEGF inhibitor and frequently had also failed an immune checkpoint inhibitor. And all the patients with the black arrows were still doing well on therapy at the time of this analysis, and the yellow star patients had achieved a RECIST partial response. So based on these promising data, this drug has gone to phase 3 testing. Now, another approach to dealing with on-target versus off-target effects, if you can't do a formal rescue by engineering or re-engineering the target, is to restore some critical downstream effector function and see if that rescues. And so, for example, there are, as I mentioned, a number of cancers, including leukemias and gliomas that have mutations in IDH1 and IDH2. This leads to the accumulation of millimolar amounts of R2HG, a so-called oncometabolite, that then perturbs various enzymes that normally use 2-oxyglutarate, a related chemical. Now, Julie Lossman in my lab, when she was in my lab, had an interest in IDH mutant leukemia cells. So she generated an isogenic system where she has leukemic cells that are wild type for IDH1 or have this canonical IDH1 mutation. She then treated these cells with a tool compound developed by Agios that blocks 2-HG production. And you can see that indeed, this compound does lower 2-HG levels. Julie had also shown that when the cells have the IDH mutation, they acquire the ability to proliferate even in the absence of cytokines, in contrast to the wild type cells. And so what Julie could now do was to treat the mutant cells with the Agios inhibitor, and she saw that indeed she suppressed their proliferation. But again, this is a down assay, a decrease in the accumulation of this metabolite. This is a down assay, decreased proliferation. So maybe this is just a noxious poison at this concentration, and maybe this is wholly nonspecific. But Julie was able to convince herself that this was on target because on the right, she's doing the same experiment, but rather than giving the cells the IDH mutation, she's giving the cells an esterified version of R2HG provided to us by Ryan Looper that enters cells and then gets trapped at tumor-relevant concentrations. And here you can see these cells are now insensitive to the drug that blocks 2HG production, presumably because you're already giving them the 2HG. And thankfully, now we have an inhibitor of both IDH1 and IDH2 that have been approved for the treatment of leukemia based on the pioneering work of the scientists at Agios. Now I'm going to switch gears and briefly talk about the story of an infamous drug called thalidomide. You may know that this drug was available over the counter in many countries back in the 50s and 60s of the last century. And unfortunately, when women took this drug during pregnancy for morning sickness or insomnia, they had a high risk of having children with these horrific limb defects. And these came to be known as the thalidomide babies. But then through sheer clinical serendipity, it was discovered that thalidomide and thalidomide-like drugs, such as lenalidomide, are really wonder drugs for certain B-cell malignancies, and in particular, multiple myeloma. And that's probably, so now we had this infamous drug that causes limb defects, but now it's a wonder drug in cancer. How does it work? The foot in the door came from Hiroshi Handa, who immobilized thalidomide on a Cephros bead and recovered a ubiquitin ligase that contains cereblon as the substrate recognition unit. In their paper, in their final figure, they did a number of down assays that suggested that thalidomide was probably a cereblon antagonist, that thalidomide inhibited the cereblon ubiquitin ligase. And that's maybe where things would have stood, but then clinical investigators noticed that in patients with multiple myeloma, frequently when their myeloma cells stopped responding to thalidomide-like drugs, their myeloma cells often were no longer producing cereblon. Conversely, high levels of cereblon tended to predict for responsiveness to the IMiDs. So the people who wrote these papers all said, well, then this provides further evidence that cereblon is the target of the IMiDs, which is true. But no one then said, and it can't be an antagonist. It can't be a simple antagonist, because if it was, then loss of cereblon should basically phenocopy the drug. It should be a form of cellular suicide. So again, the power of logical thinking. So this suggested to our group and Ben Ebert's group that cereblon, once bound to a thalidomide-like drug, acquires the ability to destroy a myeloma oncoprotein. And so to look for that protein, we devised a high throughput screen for that would allow us to monitor changes in protein stability. So you have the protein of interest is fused to firefly luciferase, and then off the same transcript, we encode Ranilla luciferase. And now the firefly to Ranilla ratio can be used as a surrogate for changes in stability of the protein of interest. So the idea would be to treat with your perturbin and then measure the changes in the firefly to Ranilla ratio. And I should point out that this was somewhat influenced or motivated by very lovely work that my colleague Steve Elledge had done at Harvard with fluorescent proteins. So Gang Liu in the lab, when he was in the lab, made a collection of 12,000 different protein open reading frames in that vector. So each well has a different open reading frame fused to firefly luciferase. You then treat with lenalidomide or vehicle and look for examples where lenalidomide is destabilizing or stabilizing the protein of interest. And of course, we were particularly interested in examples where lenalidomide might destabilize a protein. So this work is published, but it turned out two of the true positives were IKZF1 and IKZF3, which are two B-cell transcription factors. These validated in low throughput assays. So here I'm showing you cherry picks where you can see that the IKZF1 and IKZF3 fusions are downregulated if you look at the firefly to Ranilla ratio. This is specific because these three paralogs were not affected. And this was not an artifact of being fused to firefly luciferase, because here instead the transcription factor has been fused to an HA tag, and you again see downregulation. Now, it was certainly plausible that IKZF1 and IKZF3 were the relevant targets, but to establish causality, Gang made 3 hairpins against IKZF1 and 3 hairpins against IKZF3. Now, I'll point out for reasons we still don't understand, when you knock down IKZF1, you knock down IKZF3 and vice versa. IR4 is known to be downstream of these transcription factors. And on the left, I'm showing you a competition experiment where we mark in red the cells that have the hairpin against IKZF1 or IKZF3, but we mix them now with cells that are not RFP positive and they have a control hairpin. So you see very nice dropout of the cells that have the hairpins against IKZF1 and IKZF3. And I should point out, this is an IMiD-sensitive myeloma cell line. But again, this is a down assay. You know, I bet there are many expressed genes that when inhibited or downregulated would adversely affect the fitness of these cells. So this is certainly consistent with IKZF1 and IKZF3 being the IMiD target, but we wanted an up assay. Now, the way to make an up assay here was to appreciate that IKZF2 is insensitive to lenalidomide, and there are only 7 amino acid differences between IKZF2 and IKZF1. And so Gong toggled each of these one at a time until he figured out what the critical residue was. And that allowed him to make drug-resistant versions of IKZF1 and IKZF3 that are now completely resistant to lenalidomide. And now he could do rescue experiments. And you can see these cells now become insensitive to lenalidomide. Now, this piqued our interest, and I think a lot of people's interest in small molecule degraders. And so we wanted to think about, could we go back to HIF2 alpha and find degraders for HIF2 alpha? And so we did some screens for potential HIF2 alpha degraders. And in one of the early screens, we identified a famous or infamous compound called YM155 that actually does downregulate HIF2 alpha. But again, just to illustrate why we hate down assays, despite the fact you see figures like this all the time in the literature, this is actually a sleight of hand because, of course, vinculin, the loading control, has a long half-life, whereas HIF2 alpha, even in cells lacking the VHL protein, has a short half-life. So, you know, I could do this with cyclohexamide. I could do this with actinomycin D. So again, you would need many more controls to convince yourself that this was a specific effect. And so we wanted to develop an up assay for protein degraders, which is what Sagar Kaduri did in the lab. So here, the protein of interest is fused to a modified suicide gene. It's actually a modified cytidine kinase developed by Jeff Medin. This modified cytidine kinase accepts a non-natural nucleoside and converts it into a toxin. And then there's also a GFP off the same transcript. So the idea here is to add your chemical or genetic perturbant, add BVD, or now look for green survivors. So here's a proof of concept experiment where we have cells engineered to have the IMiD target fused to the suicide gene. Immediately, you can see the 2 wells that had pomalidomide, an advanced IMiD. Over here is an assay positive that simply interferes with uptake of BVDU. And so we've now used this screen to look for multiple degraders of high-value undruggable proteins. So in closing, this is a famous picture in clinical oncology. You may recognize this is a patient with malignant melanoma who had a BRAF mutation. We talked about that earlier. They're treated with a BRAF inhibitor. They have this Lazarus-like remission, but then shortly thereafter, they develop resistance and they're right back in hot water. And this has led to a lot of hand-wringing in both the scientific press and the lay press as to whether targeted therapy is really all it's cut out to be. And here's Jim Watson saying, given the seemingly almost intrinsic genetic instability of many late-stage cancers, we should not be surprised when key old-timers in cancer genetics doubt being able to truly cure most victims of widespread metastatic cancer. Now, I would argue Jim is, again, falling prey to this fallacy I told you earlier, the multiple mutation problem. But I think more importantly, he's forgetting perhaps why we used to teach in the 70s and 80s why curing cancer would require combination chemotherapy. So it doesn't require combinations because N mutations equals the need for N drugs, but it does require combination chemotherapy to prevent resistance. So imagine a cubic centimeter tumor. A cubic centimeter tumor might have 10 to the 8th to 10 to the 9th cells. The tumor burden in most cancer patients is more like 10 to the 10th to 10 to the 12th cells. So even if you had a great drug, but 1 in a million cells were resistant on a genetic or epigenetic basis, then the chance of cure with one drug is essentially 0. But the trick is to make the math work for you. And so the classical solution is to combine drugs that have distinct mechanisms of action. And because they have distinct mechanisms of action, you hope that they're not going to be cross-resistant to one another. And you hope their toxicities won't overlap in a prohibitive way. Because then in a perfect world, if you had 3 drugs where each drug is a 10 to the -6 probability of being resistant, now the chance of a cell being resistant to all 3 drugs is now 10 to the minus 18th. And now the math works for you. And we know this works because this is what the treatment of tuberculosis looked like in the United States before the advent of combination chemotherapy. It also arguably is what's allowed us to control HIV. And frankly, I think it is why we do cure patients when we do cure patients with combination chemotherapy. And here's actually a nice study from Adam Palmer and Peter Sorger, where using a variety of genetic screens and mathematical modeling, they were able to make a very convincing case that the reason R-CHOP is curative and non-Hodgkin's lymphoma is almost certainly because we're combining drugs with distinct mechanisms of action. Now, I showed you this a moment ago. These were the swimmer's plots in patients with advanced kidney cancer treated with that HIF2 inhibitor, belzutifan. But a truism in medical oncology is that most drugs work better if they're used in a frontline setting rather than a late line setting. So we were able to convince Peloton and then Merck to treat 61 patients with von Hippel-Lindau disease who had measurable kidney tumors who had never been treated medically before. So you may know that patients with VHL disease have actually inherited a defective version of the VHL gene from mom and dad. And so there is increased risk for a number of tumors, including kidney tumors. And because they develop so many tumors, they're often put in careful surveillance programs. In fact, many of these patients have been followed by Marston Linehan and his team at the NCI. So now here's the same swimmer's plots. Here's 1 year. You can see most of the patients are doing well. Frankly, I think the response rate is now up to 60%. And based on rather extraordinary results in the phase 2 trial alone, this drug has now been approved for the treatment of VHL disease-associated neoplasms. But just to give this sort of a human face, here's a social media post that was shared with me. This is one of the patients who was on the trial. You can imagine these patients have had this sort of Damocles over their neck their entire life, knowing how this disease has ravaged their family generation after generation. But here's a patient saying, I never thought I'd see this day. And they're describing how their tumors were getting smaller, were stable, or in some cases had disappeared. Here's a vlog that a patient shared with me, and I have permission to show their face.
Hey everybody, it's Justin. I just wanted to give you a quick update. I am in a gondola right now in Taiwan. Over there is Taipei 101. The gondola is actually right by the Taipei Zoo. But I just wanted to give you a quick update and say I'm doing well. I'm enjoying my trip. If it wasn't for the PT2977 drug trial, I would have never been able to come out here and do what I'm doing right now. So I just want to thank Peloton, and I hope Merck will fast track this drug for a VHL treatment. So if you guys are listening, hopefully you guys will put it on the market to help VHL. But yeah, keep watching these videos. I'll be making more, and I'll get better at it. And I have to get the angles right because I kind of look fat, you know.
Yeah, I love that, because at that age, you should be more concerned about whether you look fat on your vlog than whether your next MRI is going to be horrific and you're going to be told to get your affairs in order. So I'll close with my required reading. Many things we talked about today are encapsulated in this review article I wrote called Common Pitfalls in Preclinical Cancer Target Validation. And I wrote this piece as a tribute to my late wife, who was a breast cancer survivor, but she then died of a glioblastoma in 2015. And one of the things that was quite clear as she was going through her treatments was that everything we had available to treat her was based on science that was done 10 or 20 years ago. And frankly, if God forbid one of our children or grandchildren or loved ones gets this disease in the future, they're counting on the science we're doing today. So I can tell you from experience, it's not about fooling reviewer 3. It's about whether what you publish is true and robust and can be built upon by others. And if you don't care about that, just step out of the way, because you're becoming an impediment. You're becoming a dominant negative. So all of us, and I start with me, have to figure out a way to raise our standards so that more and more of what we publish can be built upon by our colleagues. So with that, I'll thank you very much, and I'm happy to take questions.
Thank you very much, Dr. Kaelin, for a wonderful presentation. Okay, so I don't see questions in the Q&A box. So please, anyone who has questions, type in. Okay, so I see one coming in. This is from Sam Patnaik from NCANS. How did you generate so many proteins with the luciferase tag? He's asking.
Yes. So we took advantage of recombination-based cloning. So this was done And there are several versions of this now, but we basically had a library that was created by Mark Vidal and colleagues at the Dana-Farber, where he had all these cDNAs in so-called donor vectors. And so we built a recipient vector where all of this could be done by rapid recombination cloning.
Thank you, Dr. Kaelin. So the follow-up question from Sam is, thanks. What do you think about inactive chemical controls?
Well, again, you can't have too many controls. The problem with quote unquote inactive chemical controls is a priori, it's impossible to know whether in addition to detargeting the intended target, whether you've also detargeted the off targets, because almost by definition, it's the off target you don't know about that you worry about the most. So I think it's certainly nice. For example, I was taught many years ago the power of enantiomeric controls, or I think in addition, you could have subtly altered versions of your chemical. But again, the real issue is a priori, you don't know whether the chemical change has also led to detargeting of the off target as well as detargeting the on target. So I think it's useful, but I think the genetic experiments, including the rescue experiments, are what really help the most.
Thank you, Dr. Kaelin. All right, so the next question is it preferable to try to convert down assays into up assays when possible, or to validate hypotheses generated from down assays via more specific up assays?
Yeah, I'm not sure one's preferable to the other. One thing that we have found over time is, first of all, often, not always, but often you can think of an up assay, but it often requires you to be a little bit more clever than would have been the case for the down assay. The down assay, especially for those of us who have been doing cancer biology and cancer pharmacology for years, is often almost a knee jerk. But if you force yourself to say, no, I'm going to try to come up with an up assay, you often can, but it often requires that you be a little more clever. And the geneticist Seymour Benzer had a saying, which I like, which is a good experiment should be pretty and witty. So sometimes you have to be a little witty to come up with the up assay. But I think depending on the question at hand, I think you could certainly defend in some cases doing hypothesis generation, as was said, using a higher throughput down assay, and then come up with a up assay or some validation assay that you're going to do downstream. But I have to say, having done both, whenever we can, we try to move the up assay up front, because even with relatively well-controlled down assays, you just wind up getting so much junk, at least in my hands, that you then wind up sorting through. But again, I think it really depends on the question at hand, what makes the most sense.
Thank you very much, Dr. Kaelin. Any more questions? I'll ask one until we get more questions coming in. As I listen to you talk, Dr. Kaelin, I keep thinking, what can we do? I mean, there's obviously there's something wrong with the way we're doing research now. And what sort of tangible things that we can do at the level of the infrastructure, higher level of the infrastructures as well, when it comes to journals, grants, to make a difference and change this towards positive?
It's a great question, and I'm not sure I'm going to satisfactorily answer it in a few minutes. But I do think what's happened is, in the course of one generation, when I was getting started, the pendulum was all the way in the direction of the basic science and fundamental science. That was really the really gold standard. And people who did translational research, that was frankly almost considered a second class science, partly because it's so hard, right? Because you can't control as many variables when you're doing translational research as opposed to studying a purified protein in a test tube, or maybe studying a very simple tractable organism like a yeast or a fly. So I think over the course of a generation, the pendulum now is everyone feels like they have to do translational research, and that's a science too. But I think we have to kind of get the pendulum back towards the middle, because I do think too many young people get the message that their work is not valuable unless somehow in the last figure it smacks of being translatable. And just as you need a certain training to do basic fundamental work, you need a type of training to do translational work. I just think we have to get back to understanding that everything is rooted in fundamental science, and then on occasion we learn enough, we acquire enough knowledge that we can start to see a path to translation. But I think right now people are just trying to take shortcuts to get to that translatable figure 6. So I think all of us as reviewers, as well, and I mean reviewers of papers, reviewers of grants, I think we have to take a deep breath and say not everything has to be translatable. Let's start with the science being well done, and be a little more Catholic about what will eventually be important and not be important. But I do hope, and it's so sort of sobering that maybe the most important paper I'll ever write was that Common Pitfalls paper. You know, I've had really wonderful feedback from a number of people saying, you know, if we could get closer to what you outlined there, we'll probably be better off. And that's why I recommend it as reading for young people entering the field.
Fantastic. We can circle that paper around with our attendees after the talk, after this as well. So thank you so much, Dr. Kaelin. I don't see any more questions coming in. So this was a fantastic opportunity for us to hear about these pitfalls. And we appreciate you being here today. Thank you, everyone, for attending this. I hope you found this educational. And then I hope you all join us with our next webinar coming up in a few months. Thank you very much, everyone. You all have a wonderful day. Thank you.